Hello, my name is Professor Matthew Schmidt, and I'd like to welcome you back to genetics. This whole section we've been talking about, as you can see, mutations and the nature of the gene. We're trying to really get at what a gene is. And if you're following along in order, in the last session we focused mostly on the function of a gene in the sense that we know that one gene is responsible for making one enzyme or one protein. Certainly, we've said things about the structure of the gene in the past, but we really want to get at it. We're pinpointing it more and more and more. And in this session, we're going to use mapping of viral genes, fine structure mapping, in order to really elucidate what a gene is from a structural point of view. If this seems, I know you guys know structure and function really are inseparable. One determines the other, one demands the other. So if you're a little hazy on, where we're going right now, I can promise as much as I can that it'll come clear very shortly. So the first thing we need to talk about in order to do this is just the general idea of mapping viral or in this case phage genes. In case I think we've said it before, but a bacteriophage is a virus that infects bacteria. They're reasonably easy to work with because you can do it all in culture in the lab. And there are many reasons you might want to map it. Sometimes you just want to map it to get the map. But in this case, we're going towards a structural definition of the gene. So first of all, the whole idea of mapping, we've done it so many times in so many different concepts. I say here it's based on the same way that we've always done it, which is looking for recombinants and figuring out, based on the percentage of recombinants, how far apart two loci are. I say here, just sort of like we did with flies or whatever we did it with. But with phages, we have to be a little bit more clever about it. How could one even know phages are there? You can only see them with an electron microscope. We're not going to use that. What we basically do to, quote, grow phages is to use a so-called lawn. A lawn of bacteria just means heavy growth. Sometimes it's called confluent growth. They call it a lawn because you could think of just a nice lush grass, unlike the one outside of my house, but regardless. So you have this lawn of bacteria, and when phages are growing there, what they're really doing is destroying the bacteria. In fact, bacteriophage means bacteria eater. They don't really eat them, but they do destroy them. So you get these so-called plaques. What a plaque is a clear area on the lawn where the bacteria have been killed, so they're no longer there. You can literally see a hole on the lawn. So that's the way that we can detect them. Now, certainly viruses don't have sexual reproduction, which is usually demanded for this type of mapping, right? But we can, in a very real sense, cross phages of two different genotypes by doing a process called co-infection. All co-infection means is this bacterial cell that's going to be attacked, we're going to put phages of two different types in there. And if the phage DNA that's of one type gets in that cell, and then we have phage DNA of another type, they can both get into that cell. And the two DNAs can commingle, and crossing over can occur. It's not sex, but it's simulating that type of a situation, at least in the sense that you have two chromosomes, if you want to call them that are in a position to exchange parts and to undergo crossing over. So co-infection, it's good to know that term. You're infecting the same bacterium with phages of different genotypes. And as I just said, once inside the cell, meaning once the DNA goes into the cell, crossing over can occur between viral genomes. So let's look at an example of that so we get used to doing it. Phage mutants, bacteriophage T2 is a very famous one that's a lot of work has been done with. The T-even phages, T2, T4, etc., are among the most widely studied. So here's a situation where we're going to look at two different loci, two genes of the virus. One's called H and one's called R. Now, the wild type for H, this has to do with, and don't worry about why, there are many, many strains of E. coli, but The wild type can only infect this strain called strain B, all right? But this H minus mutant can infect not only strain B, but what's called B slash 2, all right? So that's the difference between the wild type and the H minus mutant. R is another locus, and the wild type for R, small fuzzy plaques are produced, and R minus is a mutant, produces large distinct plaques. Again, for right now, you don't have to worry about why. But the point is, while these are not traits like eye color and things like that, these are things that you can detect if you're looking at bacterial growth. You can see which strain gets infected, and you can see the type of plaque that comes about. All right? So let's take a look at a cross. I know it may sound odd, but we really are sort of crossing two viruses. So one is H minus R plus, one's H plus R minus. And you can go back and look and see what, in other words, if you just did if you infected bacteria just with that, it would have the wild type trait for R and the mutant for H. The opposite if you did a single infection with that one. But we're doing a co-infection, so DNA of both these types are going to be around. So how you do it, you co-infect bacteria, and you do it at what's called a high MOI. And MOI stands for multiplicity of infection. It just means you're putting so many viral particles in there that there's no doubt that a good amount of all the types, in this case both types, are going to get inside. Now, this term lysate just means where you see the plaque, it looks like there's nothing there, but in fact there are viral, I almost said viral cells. That would be a no-no. There are viral particles there, right? So the lysate just means you're collecting liquid in that area. Lysate is the place where the bacteria were lysed. So we know that there are going to be phage progeny there. And some of which, we're assuming, are going to be recombinant because crossing over has occurred. Then check out, this is so clever what we can do. Plate the lysate onto another lawn, but it's going to be a mixture of strain B and that strain B over 2. And on that type of a lawn, 4 plaque phenotypes are going to appear. So the 2 parentals, if you just did a single infection, this is the way they would look. So H minus R plus, now this is, stick with me on this, I think it'll make sense. Small and distinct. Why is it small and distinct? Well, the R plus, remember, if we go back just for a second, R plus means small plaques, and that's what we're getting here. So the small plaques come from the R plus. The H minus, they're distinct because this is a mixture of the 2 strains of E. coli. H minus can grow on both of those strains, can infect both of those strains. So all the bacteria are getting killed in a particular area. That's what we mean by distinct, not cloudy. You really see a plaque there. The other parental, H plus R minus, the R minus is responsible for the large size of the plaque, but also if it's H plus, what we mean by cloudy here, the reason it's cloudy is because on this lawn, the wild type with respect to H plus, let's just go back and look. H plus can infect strain B only. So if you do this correctly and think of a lawn, I don't know, with 2 different grass types on there, only one of them is being killed here. So even if there's full death of all the strain B, you've only thinned out the bacteria by a half. So you see the plaque, but it looks cloudy because there is still some growth there because only one of the strains was able to get killed. All right? So you can see a small and distinct plaque and a large and cloudy plaque. Know that those are parentals. If you're looking for recombinants, you could predict what they would be because H plus R plus would be one recombinant and H minus R minus would be the other recombinant, right? And generally speaking, when one's created, the other will be created as well. And you can just sort of mix and match. If it's H plus R plus, that's fully wild type, which means small and cloudy, right? If it's fully mutant, we're going to have large and distinct. It may seem a little bit of an abstract idea, but if you know what you're doing and you look at these plates, you can definitely tell these types of plaques apart. Okay? Then, I hate to say it's simple, but you just do an analysis like we always did. Basically say, how many recombinants are there, whatever number it is, and divide that by the total, and you will get the percent recombinants, which is equal to a map distance. So here are some, I made up these results just to make the math easy. But if you see 49 of one type of parental, 49 of the other type, one each of the recombinants, the bottom line is that the recombination frequency is, all this means is the total number of recombinants, right? Which in this case is 2, 1 plus 1, right, down here, over the total number of plaques, which I made 100 just to make the math easy, times 100%, gives you 2% recombination here. And that means that the map distance between the H and the R locus, if you wanted to make a little map, is 2 map units or 2 centimorgans. Remember, those are the same thing, right? So I hope that's not, the way you do it is different than what we've done before, but the way the data analysis comes out, it's pretty much the same thing, right? How many recombinants did I get over the total number of offspring? Gives us a percentage and gives us the map distance. Not so bad. So what the heck does any of this have to do with getting to the structure of the gene? Well, a guy named Benzer, and he did a lot of work in this arena and in some other arenas. He is responsible for giving us a lot of information on the so-called fine structure analysis of genes. And he used viral genes, bacteriophages, mainly because they're easy to work with. You can have tons and tons of offspring. It's easy to see them and count them, etc. So a little background. Even before the 40s, before DNA was discovered, it was assumed that genes were fundamental units of structure. But what that meant to people at that point is that meant they're indivisible by crossing over. In other words, they're not only a unit of structure, but they're the unit of recombination. If that doesn't make sense to you, think of it this way. Genes were visualized, it's actually called the beads on a string idea. In other words, if you think of, I don't know if I can draw beads on a string, like this, here, one gene, another gene, another gene. So the idea is, and I called it the bead theory here, it's been known by a few names. If this were true, crossing over could occur, like say we had 2 chromosomes here with the genes lining up. The idea was that crossing over could occur between the beads, but never within them. So the bead, which is the gene, is indivisible by crossing over. So under this idea, following it to its logical conclusion, and this was believed, a mutation changes a gene in toto. I don't know if you know that term. It just means, it's a Latin word that means completely, 100%. It's not a little change. It's one way or the other way, from one allele to another. So even if you go back to Mendel, big P and little p, they're 2 different complete flavors of the gene. You could picture different color beads. In other words, like we just drew here. That's not a pointer, sorry. So it would almost be like saying, here's big P and here's little p. One of them is just one way and one of them is the other way. Simple as that. Now, the gene would also be the unit of mutation in this view. These are all related concepts. The unit of structure, mutation, function is another thing, and recombination. We're going to see why they're not all the same thing, though, and why the bead theory is not, in fact, correct. So, along with this idea, the gene was also seen as the unit of function, but again, before, early on, that could just mean controlling one trait. Even Mendel could have said that, right? That whatever this gene is, it controls flower color. That's not altogether wrong, but we saw in the last lecture that really what the functional activity of the gene seemed to be was that it was responsible for making some kind of a single protein that then affected the function or phenotype of an organism. Okay? So we're starting with this idea. Benzer is starting with this beads on a string idea. He worked with T4 phage, which is very similar to T2, but a little different. And Benzer did a lot of work. Why I say ultimately disagreed is that it took a lot of analysis in order to get to this idea that Benzer ultimately came up with. And what he basically determined ultimately was, yeah, the gene is the unit of function, but it can be subdivided physically into linear arrays of subunits. In other words, it's not the unit of recombination or mutation. We now know, remember what time frame we're talking about here. It was right about the same time that the structure of DNA was being elucidated, but it wasn't so widely known. We now know that the nucleotide base pairs are actually the subunits that make up a gene, and that shouldn't be so shocking to you given all that we know, right? But let's look at the experiments he did, and you'll see the elegant beauty of what he did and how it really advanced our cause, DNA or not, as far as knowledge of it. So what he did was intragenic mapping. In other words, mapping mutational sites inside of one gene, which the bead theory says that doesn't exist. You can't do that, right? So the region he used is called the R2 region. There's a reason I'm calling it a region and not a gene right now. We'll get to it all, believe me. So R2 is it's a region. You could say it's a gene for now. If you're mutated in the R2 region, we call those mutants rapid lysis. Whatever that gene does, it causes them to lyse much more quickly, and that causes plaques to be large and distinct as well. I know this can get confusing because it's not exactly for the same reason that we just saw. In this case, we're not talking about just realize that this experiment, while similar to the other one, distinct doesn't necessarily mean the same thing it did before. Large and distinct means that there are, very clear edges to them, whereas the wild type plaques are small and sort of fuzzy, sort of like ragged. The bottom line is that you can tell them apart. All right? Now we're going to talk about two different strains here. Wild type phage, it's sort of opposite from before, so don't get confused. Can grow on so-called strain B, which we knew about before, and also this strain K that we're introducing here. The R2 mutants can only grow on strain B, not K. So I apologize, but this is just the way the experiments are. You could sort of get confused, so sort of think of the other ones separately from these, even though the idea behind them is similar. Okay, so check this out because it's pretty fascinating. I remember getting a little nervous when I was a student about this part. It seemed different and it seemed difficult. Try and realize that we're trying to accomplish the same thing we've been doing with mapping, but we're doing it on a much finer level now. So basically he obtained 60 independently isolated R2 mutants. That means they all have the same phenotype. They're all rapid lysis mutants, right? And the really big question he asked was, they all have the same phenotype, but are they really all mutated in the exact same way? Or can we find different, he was going against the bead theory already by asking this question, but you got to ask the question to know the answer. Are there specific mutational sites within the R2 region, within the R2 gene? At the time he thought it was a gene. You'll see why later, why this may or may not be true. But the brilliant thing he did was he said, well, look, just if these R2 mutants are the same or if they're different, if I cross them with each other, we'll find out whether they're representing the exact same physical location on the chromosome or whether they're not, right? Look at the logic. Now, he just numbered these 1 through 60, and eventually he had hundreds of them. R2 mutant number 1 times R2 mutant number 2. If that is, in fact, the same exact spot where they're mutated, you can't have recombination in that case, so you'll never regenerate the wild type. But if they really are two distinct altered sites on the gene, we should obtain a certain amount of wild type recombinants. Okay? I'll draw a little tiny picture. Here's R2 mutant 1. I'll do in blue R2 mutant number 2. The point being this. If, in fact, say this is a gene. I know I'm going back and forth between region and gene, and I apologize. For now, let's just say this is one gene. So the bead theory says, look, R2 mutant 1 is one way in in total. R2 mutant 2 is another way in total, even though they have the same phenotype, though, which is interesting. What he was asking is it possible that maybe R2 mutant 1 is messed up, as it were, mutated in this spot, and R2 mutant 2 is mutated in that spot? If it was true, it would be possible in theory for a crossover to occur between there, and wouldn't it be possible for, you follow it along this way, for you to get two wild type sites there? And if that happened, you could regenerate a wild type phenotype. If it never happened, you'd have to assume that it was always all the R2 mutants were mutated in the exact same way. So, I guess this is a little better picture, a lot better picture than I can draw. But it shows you that if crossovers occur, the idea being, like I just said, that R2-1 and R2-2, if they really are mutated in two different places, that if a crossover of the nature that's depicted here does in fact occur, this blue chromosome, what ends up being blue in my picture, is a double mutant. So that should still have the mutant phenotype, right? Whether it's broken in one place, 2, or 25, it's not working. But this should be plus plus, meaning we've reconstituted all the working parts and we're going to get wild type. Now, so if crossovers occur, we will obtain wild type phages and double mutant phages. Check out the logic of how to see this. If we plate the lysate on strain K, only the wild type recombinants can grow, right? Because look back at what we said over here. Wild type phage can grow on strains B and K, right? The mutants can only grow on B. So again, look at this idea here. If you plate the lysate, I said on strain K, the mutants can't grow on strain K. Only the wild type can. So anything that grows on strain K is wild type. And the only way that came about could be from an intragenic recombination of the type that Benzer was postulating here. Now, if you take the same lysate and plate it on strain B, remember, mutants and wild type will grow. That'll give you a number for the total number of progeny. So it's a way of separating out the mutants and the wild type in a very, very easy way. You plate them on two different strains. Only wild type can grow on one. Everybody can grow on the other. The formula for map distance in this case is 2K over B. And I hope you can see why. Whatever grows on K, I don't know, I'm just going to make it up. Well, I actually wrote down here, if 3 plaques grow on K, let's just say, we saw 3. We know those 3 are wild type, right? But what we can't see are the double mutants that also must have been generated because they're not growing. They're double mutants. But that's why we do 2K, 2 times 3, because for every wild type one we see, there's a double mutant that we're not seeing. So if you actually have 3 plaques on K, do the math like this. And 10,000 on B, that's the total number of offspring. We have 2K times B. But the formula, sometimes you do have to memorize it, but the idea is the same, right? We just want the number of recombinants. We can only see half of them, thus the 2 times. So 2K, if there were 3 of them, makes 6. And I just said there were 10,000 on B. Look what the map distance comes out to be. 0.06 map units. That's tiny. We saw before 20 map units, 15, 2, 1 meant the genes were right on top of each other. This is 0.06. This means these sites are ridiculously close to each other. And what it also means, which we'll say, but we might as well say it now, is that these are nucleotides in the same gene that are very, very close together. I mean, 0.06 map units, we'll see in a minute what this means physically, but you would expect this. In other words, between 2 genes that are far away, if there's 3% crossing over, they're not that far, they're pretty close. But inside one gene, the distances are going to be much, much smaller, and they are. So Benzer's proof, I say here. Intragenic recombination can take place. The bead theory is right down the drain there. So the gene, this is so important, structurally the gene can be separated into smaller parts, and those smaller parts are the nucleotides. This is going along with the molecular research into DNA. So what Benzer found out, the smallest distance he could possibly measure was 0.02 map units. We now know that this corresponds to about 3 base pairs. More modern experiments have shown that recombination in theory can occur even between one base pair and the next one. Okay, so what that means is, process this all together. The gene is definitely the unit of function, right? One gene, one enzyme type of an idea. But you can't say the gene is a unit of structure, because the nucleotide is the unit of recombination and also the unit of mutation. One nucleotide can change from A to G, that's a mutation. Recombination can occur between one nucleotide and the one right next to it, that's very possible. So it tells us, yes, I mean, there may be some structural boundaries to a gene, but it is not a unit of structure because that can be subdivided. With respect to function, if part of a gene doesn't work, it doesn't work. That's it, right? You need the whole thing in order for it to work. So this is really getting down to the nitty-gritty of the fine structure, as we said, of what the gene is. Now, Benzer went on to do some more work because he found that if he used deletions, he could get a lot more map information a lot more quickly. So the original thing that put him on this track was that some of the R2 mutants, when crossed with others, never yielded any recombinants. There might be a couple ways to sort of interpret this, but Benzer realized, for a variety of reasons, that these strains contained not just a change, but they had whole regions where information was missing. Deletions of either the whole R2 region or a part of it. So here are 2 deletions. What I mean by this is all of these 1, 2, 3, 4, 5, think of them as mutational sites. They might be nucleotides, but they were things he mapped, right? So deletion 1 here, it's missing everything after 15. 16 to 20 are just gone. And this one over here is missing 1 through 10 there, right? Now, this is why these are important. If a new point mutation, one he just discovered, yields recombinants when crossed, say, just if you cross a new one with D1, you get recombinants, but when you cross it with D2, you don't get any. We've now semi-localized the location of that new point mutation. It must be in the missing region because you have to have something to cross over with, right? So if a new mutation crossed with D1 makes recombinants, that means it has to be present in an area where there's something there. The deletion delineates the part where it must be because no recombinants are made there. There's nothing for it to cross over with. So that the reason he originally did this was he was getting so many mutants. He's like, I can't just sit here and cross every one of them by each other to get map distances. Let's get a set of deletions, and that way we can localize them to a certain point and then do the more fine structure mapping. A lot of questions will ask you about doing maps with deletions, so it's good to understand this. So point mutations can be crossed with a known deletion mutant to quickly localize them to a particular region. Then they can be mapped in the traditional fashion of, say we have 10 different ones that we know are in this region. Then you can cross 1 times 2 and 1 times 3 and get that very detailed map information. So in the case of Benzer, all in all, he had about 2,400 different R2 isolate mutants, and they were mapped to 308 distinct sites in that region. So in other words, some of the R2 mutants that he got that were independently isolated were in fact mutated at the same exact spot, but there were at least 308 distinct sites that he found in the middle of what previously would have been thought to be an indivisible unit, like a bead, right? Okay. Now, in the problem-solving part, we'll talk about how to map with deletions. I mean, I just gave you an idea, but I want you to be very clear about one thing. In a lot of problems, the deleted regions, I just drew the deleted region as a space, which makes sense, but it's hard to draw nothing. So sometimes the deleted regions will be drawn as lines, and we'll say, okay, here's D1, here's D2, here's D3. You have to be careful, my point being, it might be logical to think of that as the deleted portion, what's not there. But if they tell you the line is the deleted region, then even though they're drawing it out, then the line is the part that's not there. Be very careful. That could ruin a whole problem for you. So I'm saying don't get confused. I know it's easy for me to say, but it's another one of these examples where you really have to make sure what the question's asking, how the data is being presented to you, etc. So I hope this part is not too daunting. Obviously, I hope it's understandable. But the bottom line here is that we realize that while genes certainly are, structurally an important idea, they can be subdivided, and they can be subdivided into nucleotides. And when you put this together with DNA information, it becomes very, very powerful. Now, we're going to have, if you're going along in order, we're going to have the next session is going to be about, again, defining a gene based on its function. I know we had one gene, one enzyme, but you're going to have to come back to finally, I don't know if we're going to finish the whole idea, but we're going to finish this part of the idea of gene structure and function. I'll see you then.